Okay, so I would like to introduce our next speaker. So this is Dr. Pedro Folegatti. He is an infectious diseases physician who is currently a PhD candidate in Oxford. He is the lead clinician on the University of Oxford COVID-19 vaccine programme, as well as leading their influenza and emerging pathogens programme within the Jenner Institute. He's going to speak to us today on the progress of the Oxford vaccine. So I'm going to hand over to you, Pedro. Thank you.
Thanks a lot, Rachel. Many thanks for giving me this opportunity to take us through the COVID-19 program at the Oxford University. Let me share my screen. Hopefully you can all see my presentation. Good. So essentially, the Oxford vaccine is called ChAdOx1 and COVID-19, and our vaccine is based on a non-replicating chimpanzee adenoviral vector vaccine, which then expresses the full length of the spike protein of the SARS-CoV-2 virus. The vector is non-replicating because we deleted the E1 region of the gene for the adenoviruses, with an additional deletion of the E3, which then gives us an opportunity to increase the amount of antigen that can be inserted to the vector. One of the biggest advantages of using semen adenoviruses is that it avoids pre-existing issues with immunity to human adenoviruses, and the technology allows us to essentially you can replace the E1 and E3 genes with whichever targets we want to protect people against. So it works not only for SARS-CoV-2, but we can replace that with antigens from different pathogens as well. So the vaccine antigen is encoded in the viral genome, so it's not part of a structural part of the virion, which means that any adenoviral vector vaccines are structurally the same regardless of the antigen that we want to use. Viral vector vaccines are known to induce both B and T cell responses even after single vaccination. And before COVID happened, we had about 12 phase 1 clinical trials run here at the University of Oxford, of which more than 300 people had been vaccinated with ChAdOx1 vector vaccines. But the use of chimpanzee adenoviruses is not exclusive to us. So the international community has been working on chimpanzee adenoviruses for quite a while. And before COVID, we had about over 6,000 subjects of all age groups that had previously received a chimpanzee adenoviral vector vaccine with a consistent safety profile and showing strong immunogenicity after a single dose. So at the Jenner, we've been working with ChAdOx1 vector vaccines, expressing different sorts of antigens. So we've been working on influenza vaccines, on TB vaccines, most recently with Rift Valley fevers, chikungunya, and Zika vaccines. They all pretty much use the same technology, the same vector with different antigens. But I think one of the things that allowed us to move at the pace that we have during the pandemic was our previous work on the MERS-CoV vaccine, which is essentially very similar to what we're doing with the SARS-CoV-2. So we had been previously working on a ChAdOx1 vector vaccine expressing the spike protein of a MERS-CoV antigen, and what preclinical data previously have shown was that if the vaccine was given both, either intranasal or intramuscularly, it provided full protection in mice models. We also tested efficacy in NHP models, and what we could see was that the MERS-CoV vaccine was able to induce not only binding antibodies, but also neutralizing antibodies and inhibited replication in lower airways. So the vaccine was shown to be efficacious against challenge models of MERS-CoV. And earlier this year, we published our results of a normal phase 1 dose escalation study for our MERS vaccine, where what we observed was that the high dose group that were tested was able to seroconvert in about 100% of the participants, and antibody levels were maintained for about a year post single dose vaccine administration. We haven't followed up these participants for over a year. So that's the maximum amount of data that we've got so far. And the higher dose group was also able to induce neutralizing antibodies. So we then tested neutralization of the potential for neutralization of our vaccine against different strains of the spike protein. So we assessed neutralization against homologous and heterologous strains based on the vaccine, and the vaccine was shown to provide cross protection against different MERS-CoV strains, which I suppose is a relevant finding. We're hoping that this will hold true for the COVID vaccines as well. One of the key components of viral vector vaccines is their ability to induce T-cell responses. So we assess T-cell responses determined by interferon gamma ELISpots. And what we could see in our MERS vaccine study was that the vaccine was able to induce T-cell responses peaking at around day 14, but with persistent T-cell responses up to a year after vaccine administration, after a single dose of vaccine administration. So when we started our development of SARS-CoV-2 vaccines, there were a couple of issues that we had to address first. So when first vaccines were being developed back in the early 2000s against the first SARS epidemic, what we observed in other vaccine developers was that some vaccinated animals, they exhibit great severity of infection that then controls when they were challenged with SARS-CoV viruses. So, and there are two potential mechanisms as to why this could have been happening. The first one is either driven by a Th2 skewed immune responses resulting in eosinophil infiltrations of the lungs, or an antibody mediated enhancement of infection. So there was a lot of concern when we first started the SARS-CoV-2 vaccine studies here at Oxford around the potential for disease enhancement or immunopathology. It's worth just saying that any of the preclinical animal data that we had on the MERS-CoV vaccine did not show any evidence of disease enhancement. So that gave us more confidence that this wouldn't be the case for our COVID vaccine as well. So SARS-CoV-2, so COVID, preclinical studies, they should mostly address the ability of inducing not only binding, but neutralizing antibodies, given the concerns about antibody-dependent enhancement mediated disease. They should also predominantly drive a Th1 versus a Th2 response, and any evidence of disease enhancement should be assessed. So we started off back in January, early February with our SARS-CoV-2 vaccine, and we've tested it in mice first. The vaccine was able to show that was inducing antibody binding responses, not only against S1, but S2 as well. And neutralizing antibodies were also induced in all of the mice that were tested. The vaccine was also able to induce strong T-cell responses, which were predominantly Th1, so exactly what we expected to see. With collaborators in NIH in the US, we've run a challenge non-human primate study where the monkeys were vaccinated with a single dose of the vaccine and then challenged with a high dose of SARS-CoV-2. Important to say there's a huge variation across different challenge models in non-human primates, with different groups using different doses and different routes for challenge administration as well. But what we could see in the non-human primate study was essentially that the same findings were being replicated. So the vaccine was again able to induce binding antibodies, neutralizing antibodies, and T-cell responses even after one dose. So here I'm just showing the efficacy results of that non-human primate study, which essentially what we could see was that there was no evidence of disease enhancement. So clinical scores were generally lower in the group that received the channels vaccine compared to the control one, and the vaccine was able to inhibit viral replication in lower airways, as you can see from the subgenomic RNA in both the bowel fluid and also in lung tissue as well. Not so much in the nose, so we didn't quite see efficacy against replication in the nose using that model. So just a point to say that all of this data was then reviewed by the DSMB before we could start vaccinating people with our vaccine. So this study was crucial in the sense that it gave us reassurances that there was no evidence of disease enhancement or immunopathology driven by our vaccine. And here I'm just quickly showing so we've done quite extensive preclinical work in our vaccine, and one of the studies we've done was on pigs, and this was the first time that we tested a 2-dose scheme of our vaccine. And basically what we showed was that a second dose didn't do much with T-cell responses, so the T-cell responses were not significantly boosted after a second dose, but it did significantly boost antibody responses, with most importantly neutralizing antibody responses after a second dose boost, which is something that we didn't previously seen because of multiple issues around homologous ribose with viral vector vaccines. So we started our trial in April 23rd, and this is probably the largest phase 1 trial that has ever been, and we enrolled 1,077 participants. Of those, 10 received 2 doses of the ChAdOx1 nCoV vaccine 4 weeks apart from one another, and the remainder were randomized to receive either the ChAdOx1 vaccine or the meningitis vaccine as a control. And earlier this year, we were very fortunate to publish our results that again showed that the vaccine was able to induce antibody responses, with T-cell responses being induced after a single dose of the vaccine as well. But we did observe that the group that received 2 doses didn't have better not only binding antibodies, but also neutralizing antibodies. So we've run quite a lot of extensive neutralizing antibody assays, which every single one of these assays will give you a different magnitude of responses. So it's really hard to compare these assays, but essentially what we tested was the capacity of a vaccine to neutralize various viruses at different levels. So we had a more stringent assay with 100% neutralization. So this assay is essentially an all or nothing assay. And then we used some more frequently used neutralizing antibody assays based on plaque reduction and micro neutralization assays as well at varying degrees from 50 to 70, 80, and 90%. And also we tested neutralizing capacity based on pseudovirus. And what we could see was that the very small group of 10 individuals, they did mount a better neutralizing antibody responses than the ones who previously received 1 dose. Just to remind everyone that when we originally started the program, the vaccine was intended to be given as a 1-dose program. But this data then made us investigate a bit further about whether we should be considering 2-dose regime. With regards to the reactogenicity of the vaccine, so what we could see was pretty much reactogenicity was as expected for all the previous adenoviral vector vaccines. So the local reactions are by far the most commonly observed ones, with tenderness and pain around the injection site. And what we normally see with viral vector vaccines is some flu-like symptoms, so a bit of fatigue, headache, malaise. Those things are all fairly common with the use of viral vector vaccines, and we did see that the vaccine was more reactogenic than the control vaccine we used. But after seeing some preliminary data, we decided to advise people to take prophylactic paracetamol for 24 hours after they received the first dose of the vaccine. And what we observed was that most of the excessive reactogenicity was much improved by the use of paracetamol, certainly with regards of severity of adverse events. And interestingly, what we observed in the very small group that received 2 doses of the vaccine was that the second dose administered was significantly less reactogenic than the first one. So we've done a little bit more work on the T-cell responses after a single dose of the vaccine, and what we can see is that T-cell responses are driven and are targeted both at the S1 or S2 proteins, but most importantly, what we wanted to show is that the vaccine was predominantly inducing a Th1 rather than a Th2 driven T-cell responses. So the summary of our phase 1, phase 2 findings was the vaccine is safe and tolerated, and the reactogenicity was reduced by the use of prophylactic paracetamol. The vaccine was immunogenic at a single dose with both T-cell responses and antibody responses, and was able to induce neutralizing antibodies even at a single dose, but with better results after 2 doses. One of the questions some regulators were asking were about essentially what would be the safety profile of the vaccine when given to individuals who had previously been exposed to COVID. So we didn't test individuals for previous SARS-CoV-2 infection or evidence of any serological infection before we enrolled them in the phase 1 study. So we did have about 30 individuals who were seropositive at baseline. And what we can see is that the reactogenicity profile is pretty much the same regardless of whether they had previously been exposed to SARS-CoV-2 or not. So very similar reactogenicity profiles. And this is the study that we published earlier this month, which was around the phase 2 components of our large phase 2/3 study, where we essentially enrolled adults from age 18 to over 70 years old, and we assessed reactogenicity, safety, and immune responses in that age group. And was very, so we had about 560 participants that would then split between groups that received a lower dose of the vaccine and groups who received a standard dose. The older age groups were then randomized to receive either 1 dose of the vaccine or 2 doses of the vaccine, and we assessed people in 2 different age strata. So from people between 56 to 69 and those age 70 and above. And what was very was a positive surprise. What we could see was that the level of antibodies induced by 2 doses or the vaccine didn't really differ between different age groups. So the older age groups, even the over 70s, had a very similar response to, and when it comes to antibody levels after 2 doses. We did see that the lower responses were obtained where participants received 2, so when they received 1, either low dose or 2 doses. But this was very encouraging in the sense that it showed that immune responses in over 70s is pretty much very, very promising and similar to the immune responses we were seeing in the healthy adults aged 18 to 55. And the same held true for not only binding antibodies, but again for neutralizing antibodies as well. So we didn't observe any significant differences between the different age groups. And again, we've shown that 2 doses does induce greater titers of neutralizing antibodies than a single dose. And with regards to T-cell responses, so again, the peak of the T-cell responses, they happen at around day 14, and the second dose of the vaccine didn't boost those as much. So a second dose of the vaccine does significantly increase binding and neutralizing antibody titers, but doesn't boost beyond what was originally primed by the single dose. And what we could see was that reactogenicity profile, well, with the volunteers aged 18 and 55, was pretty much similar to what we had previously observed. And interestingly, what we saw was that the reactogenicity was significantly lower in the older age groups, and even lower after the second dose. One of the key important questions around vaccines is around the anti-vector immunity. So essentially we've run some anti-vector assays, and what we can see is that regardless of whether if people receive 2 low dose or standard doses, the peak of the anti-vector immunity takes place after the first vaccine, but there's no subsequent boosting of neutralizing antibody activity against the vector after the second dose. And what we could see was also that there was a negative correlation between the antibody, neutralizing antibody titers that we've seen post the first dose and immediately before the second dose, with the number of antibodies seen after the second dose. So the greater the anti-factor antibody titers, fewer the antibody from binding antibodies against the antigen was observed. But there was no correlation between the presence of anti-factor immunity and T-cell responses. And recently we announced our interim analysis on efficacy. So our primary efficacy outcome was virologically confirmed by PCR symptomatic COVID-19 disease, regardless of severity. And those participants, they need to have at least one of objective fever defined as a temperature equal or greater than 37.8, a cough, shortness of breath, anosmia, or ageusia. And our primary analysis, which is pre-specified in statistical analysis plan, so before we opened the data for the study, was around individuals who received at least 2 intramuscular doses of the vaccine, being either a standard dose or an initial lower dose. And we had 131 cases accumulated across phase 3 studies in the UK and Brazil, with a combined efficacy of 70.4%, which was really good news. So there are several ongoing studies around the globe on our vaccine. There are studies sponsored by the Oxford University, 2 of them in the UK. The Brazil study is also sponsored by Oxford, and 2 in Africa, 1 in Kenya and 1 in South Africa. These are all Oxford-sponsored studies, which are recruiting about more than 20,000 people across those studies. AstraZeneca has quite a few studies ongoing at the moment as well, with studies ongoing in the US, with an estimate of recruitment at around 40,000 people, which will include participants in Chile and Peru as well. There are ongoing trials in India, in Japan, and in Russia as well. But I suppose one of the key good news about this vaccine is that essentially we do have agreements in place for supply capacity towards 3 billion doses by the end of 2021. So it was really good that we can show that our vaccine works, and we've got supply capacity for a significant proportion of the globe. So we were really excited about that. I'll stop here.
Okay, thank you very much. Another excellent talk. So we are going to be taking questions for Pedro at the end of this first session. So please do put those questions on the chat. So Pedro, if I ask you to come back at the end of the session, that would be great.